Alright, welcome to our second installment in Chapter 10, Genetics and Evolution. Today we are going to be tackling dihybrid crosses and gene linkage. So dihybrid crosses is looking at two genes when doing a Punnett square, and then gene linkage is like a whole lot more. So gene linkage, you're going to be looking at recombination, specifically looking at how you would be able to tell if genes are linked, and just diving much further into genetics itself. Okay, so before we start, we got to look at dihybrid crosses. Now, for all of these crosses that I'm going to set up, they're going to look pretty similar. So on the left-hand side, it has your explanation. So the F0 phenotype is the parental phenotype. So when I give you a problem, it's going to tell you what we're crossing, and I'm going to put the F0 or parental phenotype there. Below that, the parental genotype. I'm going to show you the Punnett square, and then the F1 phenotypes, which is the offspring. For this cross, we're crossing two traits, the color and the surface of peas. So we're going to cross two heterozygous plants. And so when I do this, you can see that for heterozygous, they're both going to be smooth and yellow because smooth and yellow are my dominant traits. My genotype for both of them are going to be heterozygous. And when I put my Punnett square up, you can see my gametes at the top and on the side. Gametes are what you get when you make your gametes from your parental genotype, and the way you do that is the FOIL method. So you take the first letters of each of them, the outside letters, the inside letters, and then the last letters. And when you do that, you get 4 unique gametes in this case because they're heterozygotes, but you will always end up with 4 gametes. And then you fill in the Punnett square how you would normally fill in a Punnett square by dragging and dropping the gametes from the rows and the columns. From there, you can figure out the F1 phenotypes, and in this case, you're going to get a 9:3 to 3:1 ratio. So 9 of them are going to show both dominant traits, 3 are going to show one dominant trait, 3 are going to show the other dominant trait, and then 1 is going to show both recessive traits. Now, if I were to do this again, but this time I have a heterozygote and then a heterozygous for the smooth pea surface and then a homozygous dominant for the color, I would still have both smooth and yellow because I'm still showing heterozygous and they're going to show the dominant trait, but this time you can see my genotype is slightly different for my homozygous dominant trait. Now, when I make this Punnett square, the reason why it looks different is because when I make these gametes, I have my first, which is going to be capital S and capital S and capital Y. And then my second one is going to be outside, right, for the FOIL method. So first is capital, capital. Second is capital S, capital Y. It's the same thing. So even though I don't have a second column for it, in my head, I have to remember that I have 16 total offspring. So even though I don't have a specific column, I know that this column right here happens twice. And the same thing for this lowercase s, it's going to match up with the capital Y for the inside and the capital Y for the last of FOIL. And so again, even though I only have one column, I have to remember in my head that it happens twice. For this case, I have a reduced number for my phenotypes. So 3 smooth and yellow and 1 rough yellow. Now remember, I have 16 total offspring, and 3 plus 1 is only 4. So this is only accounting for a quarter of my offspring, which means I need to multiply these numbers by 4 so that I would know what my total offspring would be. So I would have 12 smooth yellow in the entire Punnett square and 4 rough yellow in the entire Punnett square. But because I reduced, you could just say 3:1. Now, I want to know more about this specific pea. All I know is that it's rough and it's yellow. I don't know its genotype, and I don't know anything else about its parents. So really it has 2 possible genotypes. It could be lowercase s, and then heterozygous for yellow, or it could be lowercase s, homozygous dominant for yellow. Because I don't know that, I have to do what's called a test cross. So for this, to do a test cross, I need to cross my unknown with a homozygous recessive plant and then look at the outcome of it so that we can look at the offspring and determine what the actual genotype of my rough yellow pea is. The setup here for my gametes, these first two represent this heterozygous option, and the second two here represent my homozygous dominant option. And you can see that these are italicized, and so is this. So that's how you can keep track. So my test cross is going to be with a homozygous recessive rough green pea. And for my outcome, it depends on what my offspring were. If I got some green peas as my offspring, I would know that my rough yellow pea would have to be heterozygous because that's the only way I could get my green peas. However, if all of them came back yellow, I would know that my genotype for my rough yellow pea is homozygous dominant because there's no possible way for me to get a green pea, so I wouldn't have any offspring with green peas. Now, that's the kind of questions I like to ask you on tests. So for this, I would do something where I give you the offspring and you need to figure out by working backwards what the genotypes are. So for this case, we're talking about Sooty, and he is like a guinea pig, and we know that he has soft fur and sharp nails. So both of those are dominant traits. Well, in one of his matings, he was mated with a rough, smooth-nailed female. Both of those are recessive, so in this case, it's like we're doing another test cross. And I know that my offspring were rough and sharp-nailed and soft and sharp-nailed. So here is our possible our possible genotypes for the female he was mated with, right? Has to be homozygous recessive, and Sooty himself. We don't know what he is, so he could be any combination of these. So what we're going to do is we're going to test all of his possibilities. So if we test this, that's right here. If we test this, that also shows up right here. And the heterozygous accounts for all of them. So really, we'll know based on the offspring which genotype he is. The female, of course, has to be recessive. And here are our offspring. Well, we know that all of his offspring were rough fur and sharp-nailed, and then soft fur and sharp-nailed. So they all had sharp nails, which means any offspring that were homozygous recessives for smooth nails, well, we can eliminate them because that's not a possible offspring that Sooty produced, which means that this is most likely his genotype. So because only those were produced, his likely genotype is heterozygous for soft fur and homozygous dominant for sharp nails. Now, this time we are mating with another female, and we have different traits that we're looking at. So this time his offspring had pointy ears and long whiskers. So of course, we don't know what he is for his whiskers, but we know that he has pointy ears. So we know that he's going to be homozygous recessive for those because that's just what his picture is. And so we're going to test cross with a homozygous recessive for both traits. Those are our options. Those are our offspring. And we know that his kids all had pointy ears and long whiskers. So we can eliminate that one because that one had short whiskers, which means that his likely genotype is homozygous recessive for ears and homozygous dominant for whiskers. Now, next on our list to talk about is gene interaction. So gene interaction is another fancy way of saying epistasis. I should say epistasis is a fancy way of saying gene interaction. So this is the idea that one gene is dependent on the expression of another. So in guinea pigs, there's a gene C that determines if color is present. And then a second gene A is only expressed if C is first expressed. So if C is not expressed or is homozygous recessive, it doesn't matter what the A is because it's always going to be albino. But if C is present, then it does matter what the A is because it could either be a Agouti or black. And you can see this better in this Punnett square here. So again, if the C's are homozygous recessive, it literally doesn't matter what anything else is in this genotype because the C's override everything and it's going to make the guinea pigs albino regardless of what the A is because color cannot be expressed. For the rest of them, as you can see, there's at least one capital C, which means color can be expressed, and then you look at the A's to determine what color is being expressed. So just a quick overview of this before we get into gene linkage and recombinants. So humans have 23 pairs of chromosomes. 22 of them are autosomes. One pair is a sex chromosome. And XX is female and XY is male. Now we're going to be talking about gene linkage, but that's not the same as sex linked. So even though they both have the word link in them, they don't mean the same thing. So linked genes are pairs of groups of genes which are inherited together and carried on the same chromosome, whereas sex-linked disorders are carried on non-homologous regions of the X chromosome and most frequently impact males. The alleles are expressed if they're dominant or recessive because it doesn't matter because there's no alternative on the Y chromosome, so if it's there, it's going to be expressed for males. So while sex-linked is important, we're focusing on gene linkage. So for linkage groups, they don't assort independently. So this is important. They're going to always be inherited together. So for this one, we're going to be crossing two heterozygous plants. But here's the difference here. For linked genes, if you know they're linked, you have to write them out differently. And you have to write them like this, which is weird and maybe uncomfortable for you because you've never done this before. So the way that you do this is you're showing that they're linked by writing these two lines. So for this, you have a heterozygous, right? Capital, lowercase, and then you're test crossing it, lowercase, 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 lowercase. So when you have this, you have your phenotypes, right? White and short are recessive, purple and long are dominant, and because this is heterozygous, it's going to show the dominant. This is a test cross, so it's going to show the recessive. When you do the Punnett square, you don't really have any options. These two must be inherited together, so you have capital P, capital L, and these two must be inherited together. They're linked. They have to be inherited together. Whereas these ones, there's only one option, right? It's lowercase, lowercase. So when you do that, you fill it in the same way, but you're losing kind of the choice that you would get in that unlinked genes that we were doing previously. So for this, it's a 1-to-1 ratio, purple long or white short. That's your only option. Now we get into the craziness of recombination. So recombination is the idea of crossing over taking place. So from that last one, what happened here is that instead of getting that 1-to-1 ratio, right, the all purple long or all white short, we had some overlap and we had some purple short show up and some white long show up. And that's not what we expected to happen. So let's talk about why that would have happened. Well, when you go through meiosis, you have these diploid cells that are heterozygous and they replicate, and then they go through prophase 1 where crossing over can occur. And when crossing over occurs, alleles are exchanged and recombinants are produced. Oh, yes, that's what we're getting into, recombination. So thank you, crossing over, for making your life way more complicated. And so what happens is that you get an extra possible gamete. The test cross can only give you recessive for both. The heterozygous can give you two options, dominant for both or recessive for both. But the recombinant can give you a dominant and a recessive that are linked now, and a recessive and a dominant which are linked now. So now you have more choice, but the thing with this is recombinants show up at a much lower frequency than what you would expect to happen. So you can always tell what's a recombinant because the amount that would show up is much lower. So let's do an example with that. So plants that are heterozygous at both loci are test crossed, and then you get a small number of recombinants that show up. So these first ones are your normal expected heterozygous individual gametes. You know that you're test crossing, so they all have to be test crossed with the only possible gamete for a test cross. And that's what you would expect to show up. Purple, long, and then white and short. However, you get some recombinants where you have this mix-up, and so you have a small number of purple short and white long show up. So this is really, really important, and we're going to talk way more about this in class, and you're going to do some examples with this, but I want you to understand how recombinants can come to be. And so this will be our last slide, and I just want you to understand how you can figure out what are recombinants and what aren't. So the genes for kernel color and waxiness are linked in the corn plant, and in a cross between a plant that is homozygous dominant at both loci and a plant that is heterozygous at both, we want to see which of the following genotypes, so which of these genotypes are going to end up being your regular ones, your recombinants, or actually impossible to show up. So here's our initial linked gene annotation, right? We have this, which is homozygous dominant for both, right? Capital, 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 capital. And then we have our heterozygous, where we have the capitals on the top and the lowercase on the bottom. Now, when we do this matchup, there's only one possibility for a homozygous dominant. It's pretty similar to a test cross, right? Because there's only one option, so it can't go through recombination because there's nothing to recombine there. But our heterozygous one can go through recombination because it can swap these two, right? Take these W's and cross them over, and then you end up with a new combination that you didn't have before, right? Now you have a capital C and a lowercase w and a lowercase c and a capital W. This is a recombinant because you don't see this in the parents. So our normal gametes, all of them are going to go with this capital C, capital W, because that's the only option for these ones. And then the normal gametes, the majority are going to have capital C, capital W, or lowercase c and lowercase w. However, we also have some recombinants where you have a capital C, lowercase w, and a lowercase c and a capital W. So these first ones that you see, homozygous dominant for C's and W's, that's a regular one. And then heterozygous for both traits, that's a regular one because that's what you would expect to see in this cross. Our recombinants look like this, where you have homozygous dominant for C's, heterozygous for W, or heterozygous for C's, homozygous dominant for W. These are recombinants. They're not what you expect to see, but are possible recombinant offspring if crossing over occurred. So looking back at this top row of potential genotypes, you should be able to pick out which are regular, which are recombinants, and which are just impossible and would never show up. Hopefully that's what you said. The blue ones show your regular ones, and they're seen here. Your green ones show your recombinants, also seen down here. And then these last two are just impossible. They would never happen because there's no way that recombination would occur and you would get those as your offspring. All right, we're going to practice this way more in class, so if this was confusing, don't worry, we're going to practice until you get it. All right, see you next time.